So what is herpes viridae? It is a double-stranded DNA virus, and there have been research conducted that have yielded information on there being more subfamilies such as alpha, beta, and gamma herpesviridae. Although there are differences, all herpes viruses have the same basic structure, which is a core composed of double-stranded DNA, icosahedral capsids composed of 162 capsimers, which surround and protect the DNA, as well as a thick layer of virus-encoded protein called the tegment, which is found between the capsid and the envelope membrane. There is also a membrane derived from the host cell that contains virus-encoded glycoproteins, which is involved in the entry and other functions targeting replication. And then in this image, we have a photo of herpes simplex virus 1 detailing the membrane, the tegment, and the virus DNA at the center with the capsid surrounding. The replication cycle. So the herpes viridae cells enter the host cell by binding the cell surface receptors, fusing their viral envelope with host membrane. Once that occurs, the viral DNA is released into the nucleus. Inside the nucleus, the DNA circularizes, transcribing immediate early viral transcripts proteins. The replication of the viral genome of the late transcripts and proteins are expressed, where the newly synthesized viral DNA is packaged into capsids, transported through the ER and the Golgi network, where they can later be released into the extracellular space of a host to infect other neighboring cells. So this image here is also detailing just the steps that it goes through to be released from the target cell into the body, where it can be able to infect other cells. So something that happens in herpes viridae, the latency, and which can also affect other diseases, but the latent viral DNA remains a circular exosome where it integrates into the host genome and does not actively replicate. So this is the latency that occurs with the herpes viridae. The maintenance of the host genome latency depends on the expression of the latency associated with proteins, but there are definitely other external and internal stressors such as UV exposure and psychological stress, which can cause the latent virus to reactivate and produce new viral molecules. The herpes viridae causes diseases too, such as herpes simplex virus 1 and 2, as well as the Epstein-Barr virus. And this image here is just showing a screenshot of the structure of the herpes virus taken through cryo-electron microscopy.
Hello, my name is Sochi, and I will be speaking on the herpes simplex virus 1 and 2. It is most commonly known as herpes and categorized into two types, HSV1 and HSV2. HSV1 being an oral type herpes that is transmitted through oral secretions or sores on the skin. It can be spread through kissing, sharing toothbrushes, or even sharing utensils. HSV2 is a genital herpes that is transmitted through sexual contact with someone who already has HSV2, and any pregnant woman with HSV2 must consult their doctor when giving birth because the virus can be transmitted to their offspring. One must note that HSV1 and HSV2 can be spread even if sores are not present. Figure 4 demonstrates the general location of HSV1 and HSV2. On the left is the oral type, and on the right is the genital type. The symptoms for HSV1 and HSV2 are mostly asymptomatic and unrecognizable. However, if any physical symptoms are blisters or any blisters surrounding the mouth, genitals, or rectum, The blisters can leave sores if they break, which is demonstrated in figure 5 and figure 6. Figure 5 is the sores, and figure 6 is the blistering that can occur within the mouth, genitals, or rectum. To be diagnosed with HSV1 or HSV2 is just based on the appearance of the lesions or any laboratory test. Unfortunately, there is no cure for HSV1 or HSV2, but there are treatments available to relieve outbreak symptoms, decrease the number of outbreaks, and shorten healing time. Medications include famciclovir, aciclovir, and valaciclovir, and warm baths are often recommended to relieve genital sores related to HSV2. Any prevention for HSV1 is to just try to refrain from sharing toothbrushes, utensils, etc., and to just consistently and correctly use condoms. Once you get HSV1, you can be infected a second time, but you are at risk in contracting HSV2. For HSV2, any prevention you can do is just to also consistently and correctly use condoms to help prevent the spread of genital herpes, but also from contracting it, and just restraining for any sexual activity during flare-ups if you do have HSV2 to help prevent the spread.
My section was what is the Epstein-Barr virus, also known as EBV. So transmission and causes would be by bodily fluids, particularly saliva or any sexual contact. And then symptoms we found in the EBV virus is extreme fatigue, fever, sore throat, head and body aches, swollen lymph nodes in the neck and armpits, swollen liver or spleen or both, and a rash. And then the figure on the right is displaying an image of how you start to develop symptoms when you're in the second stage of EBV. Treatments that we found were staying hydrated by consuming fluids, getting lots of rest, and using over-the-counter pain and fever relievers. Prevention from EBV, there is no current vaccine available to guard against infectious mononucleosis. You may help protect yourself by not kissing or exchanging beverages, food, or personal belongings with persons who have contagious mononucleosis, such as toothbrushes.
Hi everyone, my name is Ashley and my slide is continued research on Herpesviridae. The Herpesviridae family evolved millions of years ago and continues to evolve. Researchers have studied the family's evolutionary history and continue to do so. This is because most human herpes viruses do not have a cure, and the study of its evolution and branching off can provide medicine and gene therapies. In the study, comparative genomics of Herpesviridae family to look for potential signatures of human infecting strains, they utilized pan-genome analysis and were able to differentiate between the family by HHVs, which are human herpes viruses extra genome. In the study, utilization of Herpesviridae as recombinant viral vectors in vaccine development against animal pathogens, researchers studied how Herpesviridae can be used to create vaccines by using recombinant viral vectors. This DNA virus can adapt to foreign DNA and stimulate both cell-mediated and humoral immune responses. While Herpesviridae is being studied in a variety of ways in vaccine production, the relevance now is its possibility to be used as a booster to other vaccines. This can help with cellular immune responses against antigens or B-cell priming and boosting. The family of Herpesviridae includes several vaccines in medical and veterinary use.
Herpesviridae has impacted public health in a drastic way. In 2019, the World Health Organization conducted a study that estimated almost 30% of newly sexually acquired HIV infections in 2016 were attributed to herpes simplex virus 2 infections. There is no known cure for herpes, but antiviral medication can help reduce the severity and frequency of these symptoms. But as we know, HIV is a disease that also has no cure and is treated with different medications. Right now, there There are different studies being conducted to help battle the symptoms experienced and hopefully find a cure someday. But recurrent symptoms of genital herpes can be caused by psychological stresses because of the stigma surrounding the disease, which can also further progress the virus and show any symptoms. It can also impact the quality of life and sexual relationships of an individual. However, most individuals with herpes are just living with the infection and disease and the flares of the symptoms. But there right now has to be an increased awareness of the herpes simplex virus infection and its symptoms to be able to know what to look for in any sexual partners and stuff like that. There is also a need for more improved access to the antiviral medication and more HIV prevention efforts with genital herpes simplex virus symptoms as well. These are the sources that we used during our presentation, and that pretty much sums up everything that we learned about herpesviridae.